So we've considered prokaryotic cells fairly briefly. There's a few things for you to know about them, and we're going to spend really the rest of the lecture talking about eukaryotic cells. Eukaryotic cells have a nucleus, the DNA, the genetic material, surrounded by a membrane. And this is a membrane in addition to the plasma membrane, which surrounds the whole cell. So there's a membrane-bound nucleus in a eukaryotic cell. It is the defining feature of a eukaryotic cell. Eukaryotic cells are more complex than prokaryotic cells because there are other organelles which are surrounded by their own membranes, and that creates what we call compartmentalization. So the membranes that surround these other little organelles separate different functions that are going on in different parts of the cell. So it's compartmentalized in that way. And there's also a cytoskeleton, so fibers of proteins that are stretched across the cell in different ways and underneath the cell membrane and around the nucleus, the nuclear membrane, that provide support and actually also serve as little pathways, little tracks for motor proteins which pull the organelles around the cell. So the cytoskeleton is quite important. So we'll consider the features of the plasma membrane. Primarily, we're going to look at plasma membrane in chapter 5. So we won't talk about it right now, but we will be talking about it. Cytoplasm, including the common organelles that most eukaryotic cells have. And by the way, there are other organelles that are not going to be covered here. So when you start studying specialized types of cells, you'll learn about some specialized kinds of organelles. But we're going to cover the basics. Cytoskeleton, the fibers that crisscross the cell and give it support, and the nucleus. So these diagrams that come from your textbook are pretty overwhelming. I'm going to give you some simple diagrams to practice on with answer keys, and you can practice identifying the organelles that we're going to start with for our course. And so I'm not going to have you identify everything that's here or everything that's here in terms of, let me get my mouse over here, in terms of being able to locate everything in the cell, but there I will highlight. I will add some little activities, and I will walk you through those that will highlight which parts of the cell you should be able to identify from a diagram. So don't get overwhelmed when you look at this picture. This is a plant cell, by the way. Let me get my pen. This is a plant cell here. This is an animal cell. They're just using these as kind of basic examples. There's a lot of variation that can exist between cells of different species and even cells within the same species in a multicellular organism. But I'll put some practice activities up in the folder that you can work on, and I'll walk you through those in another brief lecture segment at the end. So don't get too panicked. Okay, let's talk about the nucleus first. It contains the genetic information or the material. And like I said, on Earth today, the chromosomes, the genetic material is DNA. It is believed that in the past, RNA was the genetic material, and then over time, changes happen that DNA ends up being a more stable molecule. So it doesn't surprise me that that's the way that evolution kind of worked out. Inside the nucleus, there is a region. It's not separated by any additional membranes, but it's called the nucleolus. I have heard it pronounced nucleolus, but I pronounce it nucleolus. And either one is correct. And the nucleolus is the region where ribosomal RNAs are made. I mentioned in the previous segment that ribosomal RNAs are very particular to very particular types of species, and so they have been studied and used to classify species because their RNA, their ribosomal RNA, is so unique. But the region where ribosomal RNA is made by dehydration synthesis is called the nucleolus, and it's usually described as a dense area inside the nucleus, an area that if you put stain on the nucleus will pick up some of the stain, but there will be a really dark spot or maybe two dark spots. And so the nucleolus is defined as the region where ribosomal RNA synthesis takes place. The nuclear envelope, the word envelope means membrane. It's actually two phospholipid bilayers. So not just a single phospholipid bilayer surrounding the chromosomes, a second one outside of the first one. And so there's a double protection of the DNA that's inside the nucleus. But there are passageways, pores, that allow materials in and out of the nucleus, but they can close and open. So the nuclear pores can close or open to allow or not allow materials to come in or out of the nucleus. And in eukaryotes, the DNA exists as linear chromosomes. The most, well, I can't say most. So there's a number of linear chromosomes that is typically used as one of the defining characteristics of a eukaryotic species. In humans, your body cells each contain 46 chromosomes in the nucleus of each cell, but other species have different numbers. So, for example, yeast in the diploid state has 32 chromosomes per nucleus. You don't have to memorize this. But just as an example, fruit flies have 8 chromosomes per nucleus. So the number of chromosomes varies from species to species. But within the species, all the members of that species will have consistently that same number. So all humans have 46 chromosomes in their nucleus. Ribosomes, what you want to know about ribosomes for now is that they are the organelle that makes proteins. So they are the machine that makes the cell's proteins. They are found in all types of cells. So they're one of the few things, it's that unity that all cells have ribosomes. They themselves are made up of ribosomal RNA and ribosomal protein. So when you see that word ribosomal in front of RNA or protein, it means the protein that's part of a ribosome and the protein and the RNA that's part of a ribosome. So the ribosome is this complex of all these ribosomal proteins and all these ribosomal RNAs together. In the cell, you have hundreds, if not thousands, of ribosomes, individual ribosomes. So when you look at an image of a cell, for example, here in this picture, you can see some of the ribosomes are kind of attached to the outer surface of some of these organelles, and some of the ribosomes the artist is trying to show are just free in the cytosol. But there's tons of ribosomes. In these pictures, they're just these little dots, dot, dot, dot. I'm dotting right on top of them. And then these ones, dot, 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 dot. So the ribosomes in these artist drawings of a cell are these really small dots, and there's just a lot of them. One grouping of organelles that we're going to talk about quite a bit in the course is typically referred to as the endomembrane system. The endomembrane system consists of, and you should write this down, the outer membrane of the nucleus, because there's an outer and an inner membrane, but the outer membrane of the nucleus, which is continuous with a membrane that's called the rough endoplasmic reticulum, and that one's written here, which is continuous with the smooth endoplasmic reticulum, and which is indirectly connected to what's called the Golgi. This one you'll just have to write down, Golgi, and the plasma membrane. So we had outer nuclear membrane. If I were you, I'd write it all out. Rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi, and plasma membrane. And so this is a group of organelles and compartments that works as a unit for certain functions in the cell. So the whole thing is called the endomembrane system, and there's really these 5 components of it. So let's look a little bit into the rough endoplasmic reticulum. It's a membrane that comes off of and is continuous with the outer nuclear membrane. And on the outer surface of the rough endoplasmic reticulum are ribosomes. And so those ribosomes are sort of stuck to the outer surface of the rough endoplasmic reticulum, and that's why it's called rough, because it's bumpy from those ribosomes. And the rough endoplasmic reticulum is the location where proteins are made if the proteins are meant to be released from the cell, which is called secretion, released from the cell ultimately. It's also the location where proteins are made if they're going to be sent out to another organelle called lysosomes, or I don't know why I wrote this twice, or sent out to the plasma membrane, which is the same thing as secreted. So synthesis of proteins that are to be secreted, which means released from the cell or sent out to another organelle called lysosomes. The smooth endoplasmic reticulum doesn't have ribosomes stuck on it, so it's not bumpy, so it's called smooth. And this endoplasmic reticulum, what happens inside this compartment is the storage of calcium and the synthesis, this should say the synthesis of lipids. Even phospholipids are synthesized at the smooth ER, and also detoxification of drugs. So your body doesn't really distinguish between legal and illegal drugs, but it can detoxify, which means like get rid of drugs, and that happens in a compartment called the smooth endoplasmic reticulum. So drugs. So and your body typically, drugs are things that your cells may recognize as poison or something that causes some kind of negative effect. So anyway, what we would call drugs, legal or illegal, are detoxified or turned into something harmless by the activity of what goes on inside that smooth endoplasmic reticulum. So some cells have more smooth endoplasmic reticulum, some other types of cells have less. So how much rough ER and smooth ER are in a cell depends on what the cell is, what the cell's function is. The Golgi, which is part of the endoplasmic reticulum as well, is a flat section of compartments, or a flat group of compartments. I should say a group of flat compartments. I'll get it right eventually. That is not directly attached or directly connected to the rough ER or smooth ER, but is usually shown at some distance from them. And the function of the Golgi is to sort, package, and distribute molecules. We're going to talk about the Golgi sorting, packaging, and distributing proteins as sort of a starting point for understanding what the Golgi does. Really, the Golgi is a little bit like the post office. Mail comes in. When you put mail into a mailbox, it can be addressed to go anywhere. So all that mail is brought into the post office, and then somebody, and at this point a machine, sorts it roughly into piles, and then those piles are sorted into smaller piles, and ultimately each group of letters gets sent on the correct truck or plane to get to wherever its destination is. And so that's basically what happens inside the Golgi. Molecules are brought in, all different kinds of molecules are brought into the Golgi, but then they get sorted into piles, and then they're shipped out to whatever their final destination is supposed to be, which is fantastic. I mean, if you think about it, like how in the world can the molecules inside of this compartment know or have some way of sorting different molecules out and then having them be sent out to the correct destinations? There's a fantastic biochemical series of events that happens in the Golgi that we're not going to get into, but it does happen. We're testament that it works. And but you wouldn't be alive if this didn't work. And so the materials are brought into these Golgi compartments and then sorted and then shipped back out. So that's what the Golgi does in the cell. All right, so I'm going to take a break, and on the next lecture segment, I'm going to explain the process of protein secretion, which is going to connect together all these parts of the endomembrane system and kind of create a story, a biology story. So don't get too excited.